My first role here today will be to introduce our first guest speaker, Dr. Catherine Tsilfidis. Dr. Tsilfidis is one of the world's leading basic science researchers in ophthalmology today. She's going to speak today and share some of her thoughts on vision research, both here at home as well as across the country. She's a senior scientist at the Vision Program of the Ottawa Hospital Research Institute, and she's an associate professor in our department, the Department of Ophthalmology, as well as in the Department of Cellular and Molecular Medicine at the University of Ottawa. She has an active research program which is funded by the federal government through the CIHR, as well as through the Foundation Fighting Blindness, as well as the Kimball Foundation. And all of these are very prestigious grants that she has been awarded. Her research is aimed at developing gene therapy strategy to block apoptosis, or programmed cell death, and to slow retinal disease progression. Her laboratory is optimizing gene therapy for application to human disease. So now here to provide an overview of some of the research in Canada and around the world today is Dr. Tsilfidis.
Good morning, everyone. Can you see me above the podium? I want to thank the Foundation Fighting Blindness for giving me this opportunity. It's always an honor and a pleasure for me to be able to participate in these events and to let people know about the research that we're doing. If you want to hear me start talking and never stop, just corner me after my talk and ask about the kind of work that we're doing, because I get very excited about sharing our results with everybody. Unfortunately, I've only got 20 minutes or so this morning, and I'm going to be covering both what we're doing and a little bit of an overview of what is happening across Canada. But in terms of our particular research project, it wouldn't be possible without funding from the Foundation Fighting Blindness, without the support that the Foundation has provided for us over the years. But I also want to say that I'm particularly blessed at working at the Ottawa Hospital Research Institute and working with amazing colleagues such as Bernard Hurley and Brian Leonard, who provide the clinical expertise and let us know about the diseases that we are trying to target in the laboratory. Basically, our research project is based on the premise that regardless of what type of retinal disease you have or a member of your family has, they have one final common pathway. And as Bernard mentioned, that pathway is called apoptosis, or programmed cell death. So you may be suffering with macular degeneration or retinitis pigmentosa, or you may have had a small stroke in the eye, or you may have diabetic retinopathy. All of these diseases, and basically all retinal diseases, have one thing in common, and that is ultimately at the end of the disease, the cells in your retina die by this process called apoptosis, or programmed cell death. And so apoptosis is not something which is unusual in our bodies. In fact, it helps us in day-to-day events. So, for instance, when we are born and as we're developing in the womb, we produce twice as many nerve cells as our body will need. And the ones that make the appropriate connections survive, and the others that don't make the appropriate connections die by apoptosis, and this is a natural process that occurs. Apoptosis is also very useful for helping us defend ourselves against invading pathogens like viruses. So if a virus infects your cell recognizes that is a foreign body, and that particular cell that's been infected will actually die by this process called apoptosis to prevent that virus from escaping from that cell and infecting neighboring cells. So apoptosis is a great thing when it happens and it's controlled. However, there are very many cases where uncontrolled apoptosis can create problems. And in the retina, this is one of the issues, that the cells in the retina start going through this process a lot more than they should be. And so the premise of our research then is that if we can slow down, if we can stop this apoptosis from happening, this cell death from happening in the retina, we should be able to slow down or stop the disease progression. The way we do this is through a gene called ZIAP, and this gene has previously been shown to be a potent inhibitor or blocker of this cell death. And so how does ZIAP gene therapy work? Well, gene therapy in general, you have a gene that you're interested. You take it, you put it into a virus, you engineer this virus to produce the gene of interest, and then you infect the cell. So you put the virus into the eye, and virus does exactly what a virus is supposed to do. It infects cells. And as it infects those cells, it carries in the gene that you have provided. And so in our gene therapy, we produce this ZIAP virus, we deliver it to the eye. The virus infects the cells, the photoreceptor cells in our retinitis pigmentosa model, and then it starts to produce our gene within those photoreceptor cells. So now the genes are upregulating, or they're expressing this survival factor, and we've been able to show that at least in animal models of retinitis pigmentosa, that when they start expressing this ZIAP gene, they are protected, and we can slow down the disease progression. So as I mentioned, we've looked at it in animal models of retinitis pigmentosa, and we've been able to show that not only do we preserve the structure of the retina, but that retina actually functions as well. We've looked at it in an animal model of retinal detachment, and so what happens in retinal detachment is that, again, the photoreceptors die by this process called apoptosis, and ZIAP has been effective in blocking apoptosis in that particular disease. We've looked at it in a disease which is similar to a mini stroke, and so called retinal ischemia. And we've been able to show that in that particular case, ZIAP is also effective at blocking the cell death. And so our gene therapy basically is a general way of targeting multiple different types of retinal disease and preventing the death of those cells and slowing down or halting progression of the disease. Okay, so we have success in animal models, and a lot of the success that we've had has been funded by the Foundation Fighting Blindness and the Canadian Institutes of Health Research. And so based on this early success that we had with our animal models, we applied for and got funding, again, from the CIHR and the Foundation Fighting Blindness to try and carry this to the next step, and to try and get a little bit closer to the clinic. So what we've been doing in those studies is we've been trying to figure out what is the proper dosage of the virus that we need to deliver to the retina to have the most effective outcome in terms of preserving vision. How can we target our gene therapy exactly to the cells that need it, and not have that virus escape from those cells into the neighboring cells? And we've been able to do that. Ultimately, through the Foundation Fighting Blindness, we were also able to get partner funding from the Krembil Foundation, as Dr. Hurley mentioned. And with that funding, we are going to be testing the toxicity of the virus, because any gene therapy, before it can be applied to humans, has to have a battery of tests showing that it is actually safe, as well as being effective. And so we hope to then do the toxicity testing on this theory within the next year. And so that would mean that hopefully in the next few years we will be able to then start talking to Health Canada about trying it in patients. Okay. And so that in a nutshell is basically the research that we are conducting, and as I mentioned, I'm happy to provide any details to anyone who wants to come and see me after my talk. But the purpose of my talk today is also to give you a little bit of an understanding as to what is happening with research across Canada. And a lot of this research is research that is funded by the Foundation Fighting Blindness. Okay, and so basically, in terms of retinal disease, there have been tremendous strides that have been made within the last 20 years. And first of all, I would like to talk about sort of advances at the level of genetics. And so the first gene that was identified that caused the retinal disease was identified back in 1984. And in 1984, they didn't even know what that gene was. They just knew that it was on a particular chromosome in a particular region. So they knew somewhere over there was a gene that, when mutated, caused retinal disease. And so that was 1984. And in the 30 years since then, we now know that there are at least 225 genes. And we've identified those genes that, when mutated, cause retinal disease. Okay, so that's good news, and that's bad news. The good news, knowledge is always great news. So we have a lot more information about which genes, when mutated, can cause disease. But the bad news in that is that it's not, the situation is not as simple as we would have hoped. And so, for instance, if there had been one gene that caused retinitis pigmentosa, then everyone in the world working on retinitis pigmentosa could focus on that gene, and we would understand a lot more about what it was doing, and we would understand exactly how to target that particular gene and come up with a therapy. Now we're dealing with 200 different genes that, when mutated, can cause retinal disease. And so it sort of divides our attention in some sense. But we are still we're making tremendous progress in trying to figure out how those genes are actually affecting vision and what happens when they become mutated. Because of the fact that we know a lot more about the genetics, the other good news is that now we can start to develop targeted therapies for specific mutations. So there will come a time when you will go to your doctor, you will say, I have this particular mutation, and that doctor will say, okay, well, I know then that this therapy is not going to work in your case, but this particular one is perfect for you. And so we will get to a point where everybody's treatment is going to be completely tailored for them, and we will be able to identify exactly what will work best in each individual. Okay, and so one of the things that this sort of newfound knowledge on genetics has given us is it has made gene replacement therapy possible. And so many of you may have heard about the trials that are going on right now in Leber's congenital amaurosis. And so I'll give you a very brief overview if you haven't heard. So Leber's congenital amaurosis is a childhood disease. It is primarily the retinal pigment epithelium, which is the cells right underneath your retina, right underneath your photoreceptors that are affected. And because those cells are affected, ultimately the photoreceptors are affected and the photoreceptors die, and that's what causes blindness. As I mentioned, it is a childhood disease, and so particularly devastating because these children, often by the age of 10 or so, have lost significant vision. So what has happened in the Leber's field is that there was a mutation that was identified in a gene called RPE65. And this mutation accounts for only 5% of Leber's cases. But this particular mutation was also found in a dog model of retinal disease. And so the dogs were the first ones for which the gene therapy was tried. So the gene was discovered in 1997. In 2001, they tried gene therapy in dogs, and they were able to show that a single injection into the subretinal space of a dog could restore vision in that eye and could take that animal from being completely blind and bumping into things to a stage where it could maneuver its way around an obstacle course. The first dog that was treated, his name was Lancelot. He became extremely famous. He visited Congress. He was on Good Morning America. He really made the rounds. But he was great PR for the gene therapy field because of the fact that the therapy was so amazingly effective. And with the canine models, with the dog models, they were also able to show that the therapy was very safe. And based on those studies, they did the first clinical trial in humans in 2007. And they were able to show then that in humans, a similar dramatic effect was seen by this gene therapy. First patients that were tried had advanced disease because, as with any therapy, they were nervous about trying it in patients that had considerable vision in case it didn't work, in case it caused some kind of a damage. And so in the first patients that were tried, they were just basically trying to see whether it was safe, whether it was not necessarily effective, but whether just to prove that it wasn't going to have any harmful effects. And in those patients, just a handful of patients, not only was it safe, but some of them were able to recover some of their previously non-existent vision. And then based on those trials, they have now expanded it to younger and younger patients. Children have now been recruited into those trials, and so you can imagine regulatory agencies are not often keen on recruiting children into clinical trials, but the results from these trials have been so amazing so far that they have expanded the trials and have now recruited additional patients. And so that's the first success of gene therapy, and I think based on this success, the gene therapy field is going to expand tremendously over the next little while, not only in vision disorders, but in all types of disorders. Now, there's others. So besides the Leber's studies, there are clinical trials here in Canada for a disease called choroideremia that are being funded by the Foundation Fighting Blindness and the CIHR, and they are going to be recruiting patients very soon if they haven't started already. And so exciting because there is a disease where they know what the mutation is that causes the disease. That mutation accounts for almost all cases of choroideremia. So they're going to be able to target basically the entire population and have dramatic effects. And because of the knowledge that we have about all of this genetics, there's going to be likely many more therapies coming up in the next little while. Gene therapy trials for Stargardt's disease will be starting soon. Retinoschisis and some forms of RP are already under clinical trials. Okay, and so, I mentioned, so this is where we've gone with the genetics. If you want to know more about this, there is a session later on by Dr. Tom Lee talking about the genetics and genetic testing. Okay, and so that is some of the sort of exciting research that has been happening. But the research that I've talked about so far can halt the progression of a disease and maybe be able to fix a problem that exists in a cell, but in terms of being able to restore cells that have already been lost, that's not possible with these particular types of research. There is exciting research coming down the pipe, though, that will at some point in the hopefully near future be able to replace lost cells, and that is basically stem cell research. And so that will provide hope for people that have already lost their vision and lost a good portion of the retina, but hopefully at some point in the near future may be able to replace some of that. And so stem cell therapies started to really show promise around 2006. Prior to that, we knew that there were stem cells in the retina, but we didn't know exactly what they did in the retina. We didn't know how to trigger them and how to activate them. And we weren't sure exactly what those stem cells were doing there and whether we could harness them in order to replace lost photoreceptors. But in 2006, there was a paper that came out by a group led by Robin Ali, and he was able to show that he could take cells that were not stem cells but progenitor cells, and basically what that means is that they've developed a little bit further down the stem cell line, and they are slated to become retinal cells. If you took these progenitor cells and injected them into underneath the retina, basically what they would do is they would migrate to the appropriate place in the retina and they would make the appropriate connections. And so that this was really, really dramatic results. And it was a Nature paper. And so for those of you who don't know, Nature is basically the top scientific journal in all fields. And so every scientist has the ultimate hope of getting a paper in Nature. And so this was really big news, that he was able to show integration of stem cells into the retina. And the level of integration at that point was extremely low. So they had injected hundreds of thousands of cells, but they only got 100 or 200 cells to actually integrate into the retina. And so that's extremely small. And it really didn't have any functional effects, but it was still a dramatic leap in terms of stem cell research in retinal disease. Since that paper, there have been a whole host of people that have been involved in trying to optimize this procedure. And Valerie Wallace is an FFB-funded researcher at the University of Toronto now. She was here at the Eye Institute, but she's since moved to the University of Toronto. And so she's working on stem cell therapies with the help of the Foundation Fighting Blindness. And so basically they've gone from being able to integrate 100 or 200 cells to being able to integrate tens of thousands of cells into the retina. And that means that those cells are going to have an effect on function. They will be able to have a dramatic effect ultimately. And the fact that they can integrate into the retina and make the appropriate connections is extremely good news for people who have lost those cells and have lost vision. Okay. There's another thing that I wanted to touch on, and also for people who have lost vision, and that is that there are tremendous strides being made in terms of prosthetics. Okay, so there's something called the Argus 2 prosthetic, for instance, and basically that's just a small chip that is implanted into the eye, onto the surface of the retina. And it's attached to a camera on glasses. And so the camera takes an image of what the patient is looking at and transmit an electrical signal to the electrodes on the surface of the retina. And then those electrodes can activate the cells that travel through the optic nerve to the brain. Because in many cases of disease, even though the photoreceptors may be dead, many of the cells in the internuclear layer or the ganglion cell layer that generate the cells through the optic nerve to the brain are still alive and still have the appropriate connection. So if you can somehow trigger those cells, you can still create an image. And so things like retinal prostheses are also coming down the pipeline and will offer tremendous hope for the future. And finally, I wanted to touch on the fact that there's a lot of really, really good basic science happening in the Ottawa area and across Canada that is funded by Foundation Fighting Blindness. And that basic science may not have immediate clinical implications, but our basic scientists are letting us know about things that, like, what happens? How does the retina develop? In a disease where you have a particular mutation, how does that mutation ultimately lead to the death of that cell? In diseases such as macular degeneration, why do we get abnormal vessel growth underneath the retina that cause the disease? And how can we target specific genes within those cells to stop that from happening? And so this is all still happening in animal models and sometimes in a petri dish or in a culture dish, but we are slowly making progress and learning about how the retina functions and how it doesn't function in disease. And so hopefully those therapies will also, that information will also translate into therapies at some point in the near future. And so I was a little pressed for time, and there's a lot more that I wanted to say to you that I haven't had the opportunity to. Please come and see me after this talk, and I would be happy to provide a lot more details. I just wanted to sum up by saying that Canadian scientists, like those that are funded by the Foundation Fighting Blindness, we are trying to we are involved in an international effort to try to find vision therapies, and a lot of this couldn't be possible without the help of the Foundation Fighting Blindness and people like you that support the foundation. And so thank you very much. I would be happy to answer any of your questions. I realize that we probably don't have time for that right now, but please come and find me, and I'm happy to talk to you about it. Thank you.